Hi guys, this is the physiology review for the muscular dystrophy case. What I'm going to do is I'm going to run you through the mastery questions, and then I'm going to address the physiology questions that were asked in the session. There were quite a number of them. So we'll go ahead and get started. Just a reminder, my contact information, my office hours on Friday. On Sunday, you can get me at this link. Just click on it. It should be fairly easy. I have been meeting with students that way, and it has been working out pretty well. So I would encourage you to try it. If you're studying on a Sunday and you have a question for me, just give it a shot. I think you'll find that it's pretty easy to use. Of course, I'm always available by appointment. Just email me. There are old exam questions for the skeletal muscle stuff that will be in the document that will also have the links to this video. Just as I did with the bone and calcium, the synaptic transmission questions, and a lot of test questions from the action potential that were really just review. You'll find those questions at the end of the synaptic transmission lecture. That's probably not real relevant for a review video, but for my lecture videos, of course, you can just watch the video if you want, or just read the notes. Either one's fine. So first mastery question: which band in a myofibril does not get smaller as a muscle contracts? So when a muscle contracts, these Z lines move together, and as that happens, pretty much all these bands shorten, except for the A band, which is the length of the myosin filament, the heavy filament. The heavy filament is just a heavy filament, and what's happening is the actin and myosin are sliding over one another. Well, if it's the length of the heavy filament, that length isn't going to change during contraction. The rest of these bands will change because the Z lines are going to come together. The H zone will get smaller because that's just myosin with no actin overlap. And of course, as you contract, that zone will get smaller because you'll have more and more overlap here between the myosin and the actin. And then the I band is just actin alone, and that will also get smaller as the myosin encroaches upon the Z line. There, but this band is just the length of the myosin filament itself, and that will not change with contraction. Okay. What is the order of events in excitation-contraction coupling? So you have an action potential that comes down your motor axon. It hits the nerve terminal and the presynaptic membrane here, and that causes opening of N-type calcium channels, voltage-gated calcium channels. Calcium comes in. The vesicles, which have stored neurotransmitter, in this case acetylcholine in it, will, in response to that increase in calcium, fuse to the presynaptic membrane. You'll get exocytosis of ACH into the synaptic cleft here. And the acetylcholine will diffuse across the synaptic cleft and postsynaptic membrane, which is the skeletal muscle membrane, and the area has a special name called the motor end plate, the area of the neuromuscular junction here. And it will bind to nicotinic ACH receptors. Those will allow allow sodium to come in. They also allow potassium to go out. But the net effect is to depolarize the membrane, and remember, you get a huge depolarization of the membrane called the end plate potential, and that huge depolarization is designed to hit threshold every single time you hit threshold, and that causes an action potential to be fired in this sarcolemmal membrane, and that action potential will propagate unidirectionally away from the neuromuscular junction. Okay? That action potential dips down into the T-tubule here, and what happens is you there's the DHP receptor, the depolarization sensor, or the voltage sensor, And that will change conformation in response to the action potential, the depolarization. And that directly connects, although they don't look directly connected here, that directly connects to the ryanodine receptor or the SR calcium release channel, and through a mechanical mechanism that opens the ryanodine receptor, and you get calcium release. The calcium binds to troponin C, and you get muscular contraction. Okay? And again, this mechanical mechanism, a direct connection here between these two, even though they don't look connected, and it is not the same as calcium-induced calcium release in cardiac muscle. That is different, right? And there's a nice little summary there. Okay? What is the order of events of contraction of skeletal muscle at the myofilament level? So you have ATP hydrolysis, which leads to a cocked state. That is, your myofilament is cocked, it's unfolded, and it's ready to bind to actin, but it can't because tropomyosin is in the way. It's in the groove in the muscle over the myosin binding sites, which are the black dots here. And so your myosin is cocked, it's unfolded, it's ready to unbend, it's ready to bind, but it can't because the tropomyosin is in the way. You get calcium release into the cytosol through the mechanism I just described. That binds to troponin. The troponin, but specifically it binds to troponin C, which is a subunit here. Troponin C and troponin T and troponin I interact with the myosin and the actin in such a way that and the tropomyosin, excuse me, the actin and the tropomyosin in such a way that it shoves the tropomyosin away from these myosin binding sites on the actin, and now myosin can bind, and you get contraction as that myosin head bends, and you get a power stroke. And the actin slides that way, the myosin slides that way, you get contraction. Okay? There we go, right there. And there's our binding. All right. What is different about type 1 muscle fibers compared to type 2 muscle fibers? So here is our slide for that. Remember, our type 1 are our slow oxidative fibers. Type 2A are fast oxidative, and type 2B are fast glycolytic. The slow oxidative fibers are very resistant to fatigue. They have a low myosin ATPase activity, which means they don't generate quite as much force as the other fiber types, but they do generate that force for a long time. So what this is repeated stimulation of muscle over time, and you can see the tension generated is not going down over time. This is 60 minutes, and this fiber is still going strong. Okay, high density of mitochondria because it is dependent upon oxidative phosphorylation, and of course, oxidative phosphorylation produces lots of ATP, which keeps this contraction going as well. It contains myoglobin, so we call the type 1 fibers red fibers, it is highly resistant to fatigue. The fast glycolytic fibers are sort of, the type 2B are sort of the opposite of that. They fatigue very quickly, and they are mostly reliant upon glycolysis for ATP, and that's the reason why. You don't generate quite as much ATP from glycolysis, so you run out more quickly. They fatigue more rapidly, and of course they have fewer mitochondria, very little myoglobin, so they're called white fibers. They usually produce very powerful contractures, but they're usually very brief. Okay, again, fatigue pretty quickly. And then your type 2A are sort of intermediate. They have a high capacity for oxidative phosphorylation, and although they fatigue more quickly than the slow oxidative fibers, they don't fatigue nearly as quickly as the fast glycolytic fibers do, so they're intermediate in that respect. Their contractions are faster and more forceful than slow oxidative, but less forceful than the fast glycolytic. And they have intermediate diameter and motor unit slide, capillary supply. Everything's really intermediate here. including the resistance to fatigue. All right, so what is different about normal cardiac muscle compared to normal skeletal muscle? Cardiac muscle does not tetanize. Skeletal muscle can tetanize because the action potentials are really brief. You can fire an action potential and then fire another one before even the contraction has completed from the first action potential on a skeletal muscle. So what happens is you repeatedly fire action potentials, and the force of contraction builds and builds and builds until you hit a maximal force of contraction. You can't do that with cardiac muscle because the action potential is much too long, and you're actually partly or fully relaxed by the time the end of the cardiac action potential takes place. Certainly you're partially relaxed, and therefore you can't build by firing repeated action potentials because you'll always get some relaxation before the next action potential can be fired. That's a quick review of that. Cardiac muscle operates on the ascending part of the length-tension relationship. So this is the active length-tension relationship. Your skeletal muscle operates this at the optimal point. This is where you usually start. And then if you lengthen or shorten the fiber, either one, you lose force of contraction, okay? The cardiac muscle is different. You can't stretch a cardiac muscle beyond this point without breaking it. So it operates strictly on this ascending part of the curve. This part of the curve never really appears in a normal cardiac muscle because you can't get to those lengths. So a cardiac muscle will operate right along in here, such that in cardiac muscle, every time you increase the length of the muscle fiber, you get an increase in force of contraction. And you guys remember this, it's the Frank-Starling mechanism. Increase the filling in the ventricle, you stretch the fibers, you get an increased force of contraction, you get an increased stroke volume. So that works with cardiac muscle because it won't stretch beyond this point. It only operates on this part of the curve. Does not have parvalbumin, has different isoforms of TNI and TNT, which allows you to test for myocardial infarction by testing for the correct isoform and not worrying quite so much that maybe skeletal muscle damage is causing that increase. You're detecting the correct isoform, just the cardiac isoform. Cardiac muscle has calcium-induced calcium release, and again, skeletal muscle has mechanical release. So skeletal muscle, these two, the dihydropyridine receptor and the SR calcium release channel are connected. And when this changes conformation in response to voltage, calcium is released from the ryanodine receptor due to that direct connection. In cardiac muscle, you need calcium influx through this DHPR. It's a calcium channel. And calcium influx takes place, it accumulates in the cleft, it binds to the ryanodine receptor, and then it's the calcium binding to the ryanodine receptor that causes the calcium release. That is not what initiates calcium release in skeletal muscle. In skeletal muscle, it's a direct connection. You don't need calcium influx in order for that to happen, in order for calcium release to happen, okay? Of course, cardiac muscle spontaneously contracts at the SA node, and then the action potential propagates along and causes contraction. It does not have a neuromuscular junction, whereas skeletal muscle has a neuromuscular junction. It contracts in response to a neural impulse that transmits through the neuromuscular junction. All right, there you go. Just to emphasize that once again. Okay, so this is our first question from the session. I think I'm going to stop this video here and start part 2, and we'll cover the questions from the session in video 2. Thanks.